And good morning, everybody. My name is Andrea Riaño. I'm a biology student from University of Antioquia. And today I will present my research titled Analyzing the Interactome of Cardiac Tissue in Chronic Chagas Cardiomyopathy. Okay, so first of all, what is Chagas disease? Chagas disease is a parasitic infection that poses a significant public health challenge, especially in Latin America. It is caused by the protozoan parasite Trypanosoma cruzi. The life cycle of this parasite takes place between a mammalian host and a hematophagous insect vector. That's why the primary mode of transmission is through the triatomine bug. However, the parasite can also spread to contaminated food, congenital transmission, blood transfusion, and others. But how does the cruzi cause this disease? In this case, we see how the parasite enters the human body through the feces of infected triatomine bugs. First, the parasite invades nucleated cells. Inside them, it replicates until the cell ruptures, releasing new parasites into the bloodstream. From there, they can spread and infect new cells and organs, perpetuating the cycle of infection and damage. The clinical course of the disease involves an acute phase and a chronic phase with a range of symptoms and pathologies, of which chronic Chagas cardiomyopathy represents one of the more severe manifestations. For this research, we focus on this pathology. Following the acute stage, some patients might enter a chronic stage that can develop over many years. Over time, inflammatory modulation and scarring of the myocardium gradually occur, resulting in significant alterations in cardiac morphology. These include myofiber hypertrophy and fibrosis, that can lead to significant changes in cardiac morphology and can lead to dangerous complications such as arrhythmias, thromboembolic events, and even terminal heart failure. This progression suggests that the development of the cardiomyopathy, especially in the chronic stage, extends beyond the initially infected cardiac tissue. To understand the progression of Chagas disease, it is crucial to recognize the physiological interactions between organs, which are mediated by cell-cell communication. These interactions play a vital role in maintaining the proper functioning of the body, with the heart serving as a central hub. The heart not only drives circulation to transport oxygen and nutrients to all tissues, but is also involved in intercellular communication and the regulation of key functions in other organs through the release of hormones, cytokines, and signaling factors. With all this in mind, and taking into account the physiological interconnections between organs in the human body, called interactome, we hypothesize that patients with chronic Chagas cardiomyopathy experience alterations in the specific patterns of cell-cell communication within the cardiac tissue and with other organs. From now on, I will be referring to chronic Chagas cardiomyopathy as CCC. To construct this interactome, we began by downloading 15 RNA sequencing samples from the GEO database, 7 from healthy individuals, and 8 from CCC patients. Then, to construct the expression program, we perform a differential expression analysis. And to better understand the biological processes activated in the disease in the cardiac tissue, we perform a gene ontology analysis. Okay, so our first step was to identify differentially expressed genes. These are genes that show altered expression under a specific condition, in this case, Chagas disease, compared to a control group. In this case, we identified 3,212 differentially expressed protein coding genes. From now on, all of our analyses were performed with these genes. Now, for the gene ontology analysis, we observed that the pathways related to cardiac remodeling, most of the transcripts were downregulated. As for the pathways related to fibrosis and immune response, most of the pathways were upregulated. These findings align with the known hallmarks of Chagas cardiomyopathy, highlighting the processes of tissue damage and immune activation in the disease. Based on these results, then we ask, how is the cell-cell communication within the cardiac tissue? To explore this, we filter our D genes in different ligand receptor databases, and here we identified 186 ligand receptor pairs, where the majority of the ligands fall into categories such as cytokines, chemokines, and collagen, while the majority of the receptors fall into categories such as cytokines and integrins. Then we calculated a communication score. This score is derived by multiplying the mean expression of the ligand and the receptor, following the method proposed by Armigon collaborators in 2021. For those results, here we see like a connection to our gene ontology results, where the pairs related to cardiac remodeling show a reduced communication in CCC patients, while the pairs related to fibrosis and immune response showed an increase. Following this, then we asked how is the cell-cell communication between the heart and other organs. To do this, first we identify the ligands that are secreted into the bloodstream. And then we collected RNA-seq data on basal expression and identified the corresponding receptors in epicardial and visceral adipose tissue, brain, PBMCs, liver, and skeletal muscle. Here it's important to clarify that our RNA-seq data originated from patients in the chronic stage, and we don't have comparable data for the tissues in the same stage. That's why the communication results in this point more likely resemble the acute stage. Okay, so first we wanted to determine which tissue or organ showed to have most interactions with the heart. For that, we apply some filters to the communication score. And as you can see here, the PBMCs resulted with the most pairs, indicating the strongest communication. As for some relevant ligand receptor pairs among the tissues, we found an adiponectin that is known to have anti-inflammatory and cardioprotective effects. Then we found neural pentraxin 2 and its receptor that is known to have a role in neuronal connectivity and synaptic plasticity. And finally, haptoglobin, which interacts with a receptor in copper cells. These are liver macrophages known to be involved in inflammatory regulation in the liver. Until this point, we were able to identify the ligand receptor pairs and establish the cell-cell communication. However, we wanted to implement single-cell RNA-seq data to identify the specific cell types involved in these communications. Again, here we don't actually have single-cell RNA-seq data in CCC patients, so we instead use basal expression for the heart and PBMCs. In this UMAP plot, we can see that each point represents a cell with cells clustered according to specific types. So in the heart samples, we find cardiomyocytes, as expected, but we also find fibroblasts and endothelial cells. And for PBMCs, we found T cells, B cells, monocytes, etc. Okay, so let's see how we can take advantage of this. I intentionally didn't speak too much about these ligand receptor pairs, as I wanted to show them alongside this information. For example, let's consider the interaction between vascular endothelial growth factor D and integrin alpha-4. This growth factor is known to be involved in angiogenesis. This is the formation of new blood vessels, a key process for tissue remodeling. When we mapped the pair, we found that VEGF-D showed high expression in fibroblasts, while the integrin alpha-4 showed high expression on T cells. This was a bit unexpected since this growth factor is known to interact with endothelial cells for the process of angiogenesis. That's why we might infer that the interaction of this growth factor and integrin alpha-4 on T cells might play a role of these cells in facilitating immune cell migration during angiogenesis. Now that we've seen that we can use single-cell RNA-seq data to identify specific cell types within a sample, we also take advantage of another thing, that is to quantify the gene expression across all the samples. And taking into account that the PBMCs showed the most interactions with the heart, we then quantify the expression of key receptors on the PBMCs. And as shown here, we found a very significant result that integrin beta-2 shows high expression in the majority of the cells. With this in mind, we might think that the widespread presence of this integrin in various immune cells might serve as a critical link for the immune-mediated signaling between the heart and PBMCs. Okay, to conclude, the transcriptional and gene ontology analysis revealed that there is a coordinated response between fibrosis, immune activation, and disrupted cardiac remodeling as the main contributors in the progression of the disease. Then, by identifying ligand receptor pairs, we were able to establish cell-cell communication between the heart and other organs, in which we reveal that the PBMCs show the most interactions with the heart. And finally, the single-cell RNA-seq data not only allows us to identify the specific cell types involved in these interactions, but it also provided new insights in the cellular components contributing to the progression of chronic Chagas cardiomyopathy. Okay, now, after almost a century since the disease was discovered, there is still so much that we need to understand. Hopefully, with the ongoing research and the future studies, we will be able to enhance the treatment and management of this challenging illness. Finally, I would like to thank my supervisors, Jason and Omar, for everything they taught me, my lab partners and teachers that welcomed me into the lab. Special thanks to Hernan Hoyos, who kindly taught me the single-cell analysis. He will also be presenting in a few minutes, so please stay tuned for his presentation. And thank you for the Congress committee to this opportunity, and thank you all for coming.
And my question was, I think maybe I missed the information, but what's the effect of the capture cells expression levels? What does it change of the communication that the center of the heart?
Okay, well, to this point, we were only identify the connection by identifying the ligand and the receptor. We tried to look for the pairs that were related with the tissue and maybe the pathology, but at this point, we didn't determine what happened next. So, because then we concentrated in the single cell part. But yeah, the next step for this is to get more details about what happens when those ligand and receptors interact.
Thank you so much.